Okay, so we've been talking about this scenario, and we want to talk a little bit more about how the egg and sperm is made. All right, so we've got Donald and Daisy, and we want to know if they are the parents of Jill. So we've got to dive a little deeper and look at how the genetic information that we have is split up between eggs and sperm that we make, and how that gets passed on to offspring. So we're going to take a look at Donald. Now, if we were to pull a cell from Donald's arm, let me just grab a pen. If we pull a cell out of Donald's arm and we took a look at it, what would we see inside? What kind of genetics would he have? Well, you would expect to see a complete set of chromosomes, and half of those chromosomes come from his father and half of those chromosomes come from his mother. So let's just draw 4 of his pairs, right? Because otherwise it gets too big. Okay, so let's say he's got a number 1 from dad, a number 2 from dad, a number 3 from dad, a number 4 from dad, and he'll also have the same set of chromosomes, 1, a 2, a 3, and a 4 from his mom. All right, so we're using the 2 colors to just represent mom's and dad's chromosome. So this would be his full set of chromosomes, and he would be considered diploid. Okay, so let's write that word down. Diploid means that you have di, or 2, and ploid means the number. All right, so as humans, we have 2 times the number of unique chromosomes. So the N stands for the unique chromosomes that we have. So you can see they've got number 1s, number 2s, number 3s, and number 4s. So the N for this particular scenario I've drawn is 4. And of each of those unique chromosomes, Donald has 2 copies, so he's 2N. And humans are 2N. We have 23 different kinds of chromosomes, and we have 2 of each one. So we have 46 chromosomes total. But really, it's 2 of each kind. So we've got a chapter on, if this were a cookbook, we've got a chapter on cakes, and we've got 2 copies of that chapter. We've got a chapter on cookies, and 2 copies of that, and a chapter on casseroles, and 2 copies of that. All right, and so that's the full set. And same thing with Daisy. If you looked inside her arm cell, you would see the same kind of thing. Of course, it would carry different alleles, different genetic material, depending on who they are. Okay, so now, if we want to make sperm, let's say this is Donald, we want to make sperm, are we going to look in his arm? Well, of course not. There's only one location in a male or a female where we make these sex cells, and we call them gametes. Okay, so I'll write that term up here. A gamete is just the generic term for an egg or a sperm. So in a male, gametes are made in the testes. But same thing, if you were to look inside the testes of Donald, and look at the cell that's going to make gametes, it would look exactly like this. It would have the same full set of chromosomes. But as we explored in our activity, we want to end up with half the number of chromosomes when we're finished. Why? Because the other half are coming from the other parent. Okay, so we're going to look specifically inside the testes, and we're going to see how these gametes are made. So we're going to look at that same cell. This is the cell inside of Donald's gonads, and it's going to make gametes, okay? And so it's got the full set of chromosomes. But before it goes through this process, it actually duplicates each chromosome. Now, this is probably part of the evolutionary history of this process, which in single-celled organisms, it's mitosis, where we're just making exact copies. And in order to make an exact copy of yourself, you would want to duplicate your chromosomes, or make a full copy of this recipe book, so you can give 1 copy to each cell. This process actually cuts things in half, so you don't technically need that duplicate, but it exists because of the way this process evolved. It also gives us a lot more gametes, a lot more bang for our buck. So before we get started, we're just going to make an exact copy of each of these chromosomes. Okay, and so it looks exactly the same. It has all the same genes. It has all the same alleles. It's an exact replica of each of those chromosomes. Now let me just point out for just a minute, just for the sake of labeling these. So this here that I've drawn is a chromosome. Okay, now if we add this duplicate, we call it a duplicated chromosome. It is not 2 chromosomes. Now technically it is 2 chromosomes, but when they're stuck together like that, we consider it 1 duplicated chromosome, and we call each of these duplicates sister chromatids. Okay, just kind of a fancy word for the duplicates. All right. Okay, so now let's go back to this process. Here we have our duplicated chromosomes. We are now ready to go through the division process to make these gametes. So this initial phase right here where the chromosomes are condensed, and you can see them. In fact, that's one of the things that happens in this phase, is that the chromosomes condense, and they are sitting together with their homologous pair. Okay, so I'm going to redraw this as what it would look like if it were getting ready for meiosis. So we're going to have our cells, and in this case we're going to have each of these pairs sort of clinging together with each other in the cell. So they're not necessarily all lined up in a line. I just did that to show you the two. Okay, so they should be clinging together as homologous pairs. And during this process, you're going to see large sections of the chromosome that can potentially swap places with the other. Because they're so close in their sequence, they can swap places with each other, and that is called crossing over. So you're going to see that here at the beginning of this process, crossing over. All right, so there are a few things that we need to point out that happen in this first phase. Let me label this phase. It is called prophase, meaning the very first phase. Okay, and there's a few things that happen during this phase. Number one, you see that the chromosomes are condensed, so you can actually see them. Number two is you're getting some crossing over events happening. And number three, you'll start to see some ropes forming to pull the chromosomes apart, these microtubules. We're not going to draw those in this particular picture. We'll deal more with that in mitosis. But here's prophase. So now, if we want to line up so that each of these gametes, when the cell splits, ends up with one of the pairs, there's a very specific way that we can line up that's going to make this possible. So I'm going to draw it here. If we lined up end to end as single chromosomes, how are you going to ensure that only one of the number ones goes to one side and the other one goes to the other side? It's not like the cell has eyeballs and it can look at them and say, I want that one, and I want that one, and I want that one. It has to just send out its ropes and grab the chromosomes. So if we line them up, and it really doesn't matter, I'm going to draw them like this. If we line them up as pairs in the middle of the cell, now when the ropes come out and grab chromosomes, they necessarily grab a number 1, a number 2, a number 3, and a number 4. So it's very important to take note that when they line up in this phase, they line up as homologous pairs, which is going to be different than what we'll see in a moment. Okay, and this phase is called metaphase because they're in the middle. Now, we're going to talk about this in a minute, but division happens twice in meiosis, so I'm going to label these as prophase 1 and metaphase 1. Okay, so let's keep going. So I've redrawn metaphase 1 here. We're now going to divide the cell. So you can see that if it begins to split, so I'm going to draw the cell sort of elongated because that's actually what happens. It kind of gets big. And the chromosomes are being pulled to each side. Okay, so there's our mom chromosomes. Here are our dad chromosomes. The ropes are pulling them up and down. Okay, so you can see here, what are we separating? Okay, so we are separating what here? Separate, that's right, homologous pairs. I'm going to abbreviate homologous pairs. Okay, so once we've separated them, let's go ahead and continue to separate them. As they separate, the cell sort of starts to pinch together a bit as it's beginning to divide into two. All right, so here's our 1, our 2, our 3, and our 4 from mom, our 1, our 2, our 3, and our 4 from dad. All right, so let's label these. So that was metaphase. When we begin to divide, we call this anaphase. Okay, this is anaphase 1. And then the end, telo means the end. So telo or telophase is the end here. We're starting to pinch apart. And then eventually we're just going to break. We're going to split the cell. So cytokinesis, which means cell splitting, we're going to split it into two. All right, so here's one and here's one, and I'm going to draw them. So we look on the top one, we have our number 1 from mom, our number 2 from mom, but our number 3 and our number 4 are from dad. And the bottom one is exactly the opposite. We have the 1 from dad and the 2 from dad, and the 3 and the 4 are from mom. All right, so if we look at these outcomes, these daughter cells, let's talk about them for a minute. So number one, you should see that they're sort of the exact opposite of each other. So they're going to carry the different alleles from those chromosomes, and these two would make very different babies. Now, let's talk about another thing, and that is, are they diploid? Are they still 2N, or are they somehow different? So let's start with N. How many unique chromosomes do they have? Well, they have a 1, a 2, a 3, and a 4. So their N is still 4, but do they have 2 copies of it? Well, the answer is no. They only have 1 copy of those 4 chromosomes. So they are 1N, or what we call haploid. So in meiosis 1, we have gone from diploid to haploid. Okay, but if you take a look at this cell, it is not ready to make a baby. It's not a good sperm cell. Why? Well, it still has 2 copies of every chromosome clinging together as those sister chromatids. And we can't have that. We'd have too much DNA. So we still need to split this apart one more time. So I'm going to take this cell, and we're going to draw the second split of that particular cell. The previous page, this other cell right here will also go through the same process. So you'll end up with 4 unique gametes. Okay, so here is that haploid cell. It's got number 1 and 2 from mom and 3 and 4 from dad. Okay, so now all we have to do is get rid of that sister chromatid. It is haploid, and we're at the beginning again. So we're going to call this prophase again, but in this case, it's the second time we've been in prophase, so it's prophase 2. Now here, when we line up to divide, how do we want to line up? Do we want to line up as pairs, or do we want to line up end to end? And the answer is that we want to line up end to end because there aren't pairs anymore. Okay, these are 4 unique chromosomes. So this phase is going to look exactly like the phase I just drew. We're just going to show them lined up in the middle. All right, and you might be wondering, does crossing over happen in prophase 2? No, it doesn't, because we don't have unique or homologous pairs there. They are all unique, so they're not going to cross over. So in this case, this is metaphase 2 because they are in the middle, but in this case, how are we lining up? So you should be able to answer that we are lining up end to end. Okay, so this time when the ropes come out and grab, they're just going to grab 1 sister chromatid of each of those chromosomes. All right, so again, we're going to start to divide. So I'm going to elongate this cell a little bit, and it's going to pull apart the sister chromatids. Okay, so there's our 1 and our 2, here's our 3 and our 4, and they're being pulled apart by the ropes. And notice they're identical. Now, if crossing over had occurred, you would have bits and pieces of the other chromosome and they would not be identical. Okay, and I might actually draw that in a moment. So that is anaphase, just like before, but this is anaphase 2. And then we're going to start to pinch it apart, okay, in the end phase, so telophase. And we've got our number 1, our number 2 from mom, and our 3 and our 4 from dad. And then we're just going to finish with cytokinesis again. And we'll split this into 2 daughter cells. Okay, now notice in my case, the drawing that I've done, they are identical, 1 and 2 from mom, and 3 and 4 from dad. Now let's look at these for a second. Are they diploid or haploid or something else, right? So in this case, how many unique chromosomes do they have? Well, you see a 1, a 2, a 3, and a 4. They have 4 unique chromosomes, and they have 1 copy of each. So they are still haploid cells, just like before. Now, if crossing over had occurred, you would have noticed that one of these chromatids that are now chromosomes would be slightly different. So let me illustrate that. So let's go back to this picture. These finished cells, this one and this one, are they ready to be sperm? Well, yes, they have half the number of chromosomes and they are ready to go. So we would call these finished spermatids, and they're ready to go fertilize an egg. And if you go back, so here's that cell again, and remember this came from originally, the cell had a number 1 and a number 1 from mom and a number 1 from dad. If crossing over had occurred between these, what you would end up with is something like this. And I'll just draw it like this. Can you see what happened there? So that one arm of the chromosome actually swapped places. So if this were to be the chromosome pairs that had separated to this point, this number 1, let me redraw it here actually. that number 1 would have been half blue and half red. Okay, whereas you might have seen the other one would have had the other. All right, so you're going to have uniqueness in all 4 of the resulting gametes. So let's just do a quick overview again, starting with the original diploid cell. Okay, the first thing that happens is we're going to duplicate all of those chromosomes in preparation to go through meiosis. Okay, and then in meiosis 1, what are we splitting apart? We're splitting apart each of those homologous pairs. And so we end up with 2 chromosomes that have just one number 1, one number 2, and let's just for fun make the 3 and 4 of Dad's had gone to the other one. Okay, and these can split up any way they want to, and that's what we call the law of independent assortment. Okay, so that's what happens during meiosis 1, and you end up with 2 cells that are now haploid. So this is meiosis 1. Okay, and then in meiosis 2, we simply split those sister chromatids apart. Okay, so we'll draw that. So each of those single chromosome or duplicated chromosomes are going to split apart. So we end up with this one having 1 and 2 from Dad, this one having 3 and 4 from Dad, and 3 and 4 here from Mom, and 1 and 2. Okay, and these again are haploid cells, so that was meiosis 2. And this is how we end up with, and we'll just make these sperm, gametes that are haploid.